So what we're going to do is we're going to finish up the program here with me, kind of giving my update of the 2020 season and thoughts heading into 2021. Again, at the end here, we will go ahead and post the QR code for this last hour, and then we will also go ahead and cycle through the QR codes throughout the day as well, give everyone one more chance to screen those. So, going to get right into my talk here. The number one question of the 2021 extension season I've gotten is, where is the weed guide and how can I get it? So here, if you want to scan, here's a QR code for the weed guide for the electronic version. I'll have this again at the end of this presentation. If you want to get it online, it brings you to the website where we have the weed guide. I got confirmation earlier this morning. It is finally in at Morrill Hall. So if you're near Fargo, you can go to the basement, collect it there, call the phone number on the screen, and you will be able to get access to the weed guide if you call that phone number. And then again, out the rest of the state, they will be available soon at county extension offices. So we'll ship them out from Fargo here today, and it will be available. So not as many chances this winter to hand it out in person, but they are available in person, and we have printed plenty of them. So make sure you get your copies. If you go to the back of the weed guide, we have a weed of the year every year. This year we went with green foxtail. And so Brian just shared these data sets, so I'm not going to go too much into detail with them. But Brian just mentioned all the good reasons why we decided on green foxtail. It's pretty competitive. We are getting more samples with herbicide resistance, particularly our group 1 and group 2 herbicides that we use in small grain crops. So again, this chart is pulled straight from our Weed of the Year page in the guide. So you can read more about green foxtail and look at those numbers a little bit more closely once you get the guide either in print or electronically. The other thing I want to cover, last year or 2 years ago, Dr. Peters and myself and some of our Minnesota colleagues created a quick front and back fact sheet about all the different herbicide resistant traits that we have and all the crops that we grow in North Dakota and Minnesota. So if you Google PS 1945, that's the number of the publication. It's updated again and available online. We did not print any this year, though, since, again, we don't know how many we'll be able to hand out. What I wanted to just quickly show here is here's table 4 from that publication, which is the soybean section. The main purpose of having this on here is I want to remind everyone that we did get approval for Extend Flex soybean. So we will be able to plant that soybean in the 2021 growing season. And again, as a reminder, for those who aren't aware, that is basically the Extend soybean plus resistance to glufosinate, which is Liberty. So similar to the E3 soybean, where we have 2,4-D glyphosate glufosinate resistance, now we have that for dicamba with Extend Flex, dicamba, glyphosate, and glufosinate resistance. So that kind of leads me to the next thing I wanted to touch on is the updated labels for dicamba for use in Extend soybean. And so this is our 4th attempt at labeling these products. And so the past couple times we've had a 1 or 2 year registration. The labels that we got in late October, early November, we now have a 5-year registration. So end in 2025. All the old restrictions are still in place. So everything you learned in training last year to apply these products, those restrictions are still in place for use of these products going into next year, with a few additional restrictions to think about. I'm going to briefly cover them here. We do have to go through training again to use them. So if you get trained, you'll relearn all these things I'm going through on the slide. But some of the main differences to know for these products. The first one is they are now labeled for use only in the Extend or Extend Flex soybean system up here. We don't have cotton, so I'm just focusing on soybeans. So we can't use Ingenia or XtendiMax in fallow or in corn, instances like that. Their labels are just for the Extend soybean systems heading into next year. There's now a national cutoff date of June 30th on these labels. And North Dakota, South Dakota, and then in 2021, Minnesota as well are all applying or sticking with these June 30th cutoff dates. So some harmonization between all of our states up here for that cutoff date. When we look at the products themselves, they all have that end date of June 30th. So 2 hours before sunset on June 30th is the last time we can apply these products. With Ingenia, that's the only cutoff date on the label. XtendiMax, we're supposed to stop applications after R1, which is beginning flower. And then Tavium, no applications are allowed after V4. That's mainly driven by the dual magnum component of Tavium. So a couple crop stage restrictions to think about for some of these products. The biggest adjustment for this upcoming year is an additional adjuvant that we have to add into most applications, and well, in fact, all applications for Ingenia or XtendiMax. Ingenia label calls it a pH buffering agent. XtendiMax and Tavium called a volatility reducing agent. It's basically an adjuvant that we add into the tank to reduce volatility and help mitigate some off-target movement of dicamba by corralling in some of that volatility a little bit further. So these are becoming available on the websites. Again, you have to go to the websites for these products for all tank mix approvals. And then the buffers have been increased a little bit as well. So any application, 240-foot downwind buffer unless you have an approved hood. And I didn't put it on this slide, but counties with endangered species, that buffer is a little bit longer as well. So again, all this will be covered in trainings. So switching gears, the rest of my talk, I'm going to talk about some updates of the two big pigweeds that we're most concerned about in North Dakota and the upper Midwest. And that's, of course, Palmer amaranth and water hemp have been talked about a number of times throughout the morning so far. The one thing I just wanted to show is we've always shown these shock and awe pictures of large Palmer and large water hemp from other areas of the country. 2020, we got some good pictures of 10-foot tall Palmer in North Dakota. Just less than an hour and a half or 2 hours from where I'm currently standing in Fargo. And then water hemp there's a picture within 10 miles of Fargo of water hemp as big as a truck, both in height and width. So can these pigweeds become problematic and adapted to our region? Yes, we can get these large pigweed plants up here as well. So starting just a little bit with water hemp, I'm not going to talk a whole lot of detail about water hemp, just a few things we noted in 2020 and considerations for 2021. But especially in the southeast corner of the state, the last 2 years, kind of the same set of conditions that have made them pretty good years for water hemp to thrive. Delayed planting, saturated soils at various parts throughout the summer, of course, drowned out areas, and then prevent plant, all related to water use. So I like to say that water hemp has the word water in its name for a reason. When we get these saturated soils, prevent plant, drowned out areas, we tend to find water hemp thriving in those conditions and then producing a lot of seed for us to battle in future years. So just, going with the map here of where we know water hemp is currently in the state of North Dakota. All the green counties that you see on the screen, we have confirmed water hemp. I still say the primary area where water hemp is one of the most, if not the most problematic weed, we can basically draw a line from Fargo to Jamestown and then south. That corner of the state, water hemp is one of our biggest, if not our biggest issues, but it's becoming more problematic in other areas of the state as well, all the way up to the Canadian border, finding water hemp in large numbers in some fields, found it in every county in the eastern part of the state. And then what's not on this map, but if we go not too far west of Williston into Roosevelt County, Montana, there's about a 30-acre infestation of water hemp there as well. So just because the county isn't green doesn't mean you don't have water hemp. We just have not confirmed it yet. So kind of spreading west and becoming more problematic and something to keep an eye on even out west. The one thing I want to really focus on about water hemp in 2020 was the early emergence that we saw with water hemp. So this is a picture that I put up on my Twitter May 1st, just went out to kind of scout the plots and found a lot of water hemp had emerged. Now, I know I put this on Twitter and Tom Peters and some of my other colleagues thought I was probably smoking something, finding water hemp up on May 1st. Our part of the country, we've always kind of said 3rd to 4th week of May is when we start to see water hemp emerge. Well, as soon after this picture, then I know Tom was getting flooded with phone calls and emails. It wasn't just Fargo. It was kind of all up and down the valley. We were finding early water hemp emergence in 2020. A couple of different things probably led to this. We'll have to track this more in future years. But we had that really wet fall in 2019, fully saturated soil profiles. And then when I kind of looked back and dug into the weather data, late April, we had a couple sunny, 70-degree days. So probably had some warming of that soil temperature as well on a lot of soils that did not get fall tillage or early spring tillage. So again, all that together, and maybe another factor or two that we're not thinking of right now, early emergence for water hemp. In fact, earlier than what my colleague in Iowa found. They were still about the third week of May down in Iowa for water hemp emergence. The other thing that we were able to observe is we did have a hard freeze that second week of May. If I were to rate a hard freeze for efficacy on seedling water hemp, it was poor to good, maybe poor to fair. We killed off some of these cotyledons and seedlings, but many of them did survive. So the other reason I bring that up is because this year we had a lot of phone calls late summer about water hemp and wheat. And so this is becoming more problematic in general, but couple that with early emergence of water hemp this year, later planting of wheat because of those saturated soils, and we just had a lot of escapes, particularly in the Red River Valley, of water hemp in 2020. So the pre-harvest wheat phone calls came in, pre-harvest control of water hemp, a few products to try and accomplish that, but our focus was really post-harvest and what water hemp was doing after we harvested wheat and got those fields cleaned up. So there's one picture in the bottom center part of the slide that you're seeing. One of our agronomy seed farms, I looked at one of our wheat fields after harvest. Water hemp branched out, grew, produced quite a lot of seed. So we had plants that were there, not controlled pre-harvest, regrew and produced seed as well. The other thing that we know is possible, and we definitely confirmed it, not just on research farms, but several commercial fields, is germination of water hemp after wheat harvest that also then produce seed. So, water hemp is really a plant that is, it can take advantage of almost any weather conditions as long as it's warm enough and we have enough moisture to cause germination and go to seed relatively quickly. So here's some pictures from September 18th. This plant would have germinated about the last week of August. 3 inches tall, pulled this plant out. I threshed the seed head. We found 88 water hemp seed that came out of those seed heads 3 weeks after it had emerged. So it's not just early season where we can get, early emergence and then quickly go into produce seed. We saw this late August into September. And so if this continues to be a problem in wheat and we can't control it in wheat, we're going to have to start looking at post-harvest weed control of water hemp and wheat as well. So again, was 2020 an anomaly because of that early emergence? It's possible, but something we need to keep a closer eye on going into 2021, because water hemp is certainly starting to exploit every hole in the system that we have, and it's one of our major concerns moving forward. Okay, the rest of my talk I'm going to spend talking about the Palmer Amaranth update from 2020. So here's the current map of where Palmer Amaranth is located within North Dakota. So that we're aware of. This is the North Dakota Department of Ag is the curators of this map of confirmed cases. And the different counties, the different colors in the different counties represent when we found infestations of Palmer Amaranth within the state. So the counties in blue were 2018 findings. The orange counties in that south central part of the state were 2019. And then green counties were where we found Palmer Amaranth in 2020. You'll see Benson County and Richland County are striped blue and green. And that's because we have found 2 independent infestations of Palmer in those counties, one in 2018 and then a different infestation in 2020. So the things just to briefly touch on, the 2018 infestations, with the exception of one of those, we revisited them this past summer and we're not able to find Palmer Amaranth. And we consider this a lot to the fact that these were early detections. We hand pulled and bagged and burned the plants that we found. And going back this past year, we could not find additional plants. The orange counties, those were primarily Palmer found in millet fields in 2019. Again, those were either harvested pretty early for hay or also hand pulled weeds. From there, did not find repeat plants this past summer. The green counties up in Benson, we found in some barley and soybean fields, just an infestation that was likely a year or two old. The other green counties are all related to contaminated cattle feed this year. And that's what I'm going to spend the rest of my talk talking about here and focusing on is what we discovered at these sites, and then we got our hands on some of the infested feed, and talking about some of the findings that we've had up until just about a week or two ago, still collecting data in the greenhouse. So first thing I want to show here, this is one of the, it was either Barnes or Stutzman County, one of the infestations we found there. What I want to show is, here is an entrance to a field, compacted gravel, and the only weeds that we really found here were kochia, volunteer sorghum or volunteer milo, and then some palmer. The reason I bring this up is we show these shock and awe pictures of the 10-foot tall plants, but palmer is a plant that's from the desert. It's pretty drought tolerant, just like kochia, and it can survive in these marginal field areas as well. We just won't always see it be these large plants like we're seeing, like a lot of the pictures that we see. So anywhere where kochia can germinate and survive, even if it's relatively small, basically the same thing with palmer. This particular field, though, if we just went 100 yards away, that's where we started seeing these plants that are as tall or taller than corn. And in this case, we had quite a hefty infestation of palmer at this particular location. Picture on the left side of the screen there is just kind of the wall of palmer that we found as we turned a corner looking at this field. Then if you look a little bit closer, that picture on the right, you'll see a palmer plant that's taller than the tassels of the corn. So pretty typical that we'll see in other parts of the country when it comes to finding palmer and corn, and can thrive and survive just as well here in North Dakota. Then, of course, we had to pick on a couple interns, sent them deep into the field, and the plant there on the left or in the middle part of that screen on the new picture was about a 12-footer. The one next to it was 13 feet tall. So can palmer get to be 12 or 13 feet tall in North Dakota? Yes. And it's not just the tall plants. As we kind of look, as we go deeper into the cornfield, these are a lot of the inrows. You won't see this from the road. So a lot of chest-high plants to knee-high plants we're finding in this field as well. So the palmer is growing well in the field, but we can also, the corn was mature, but a corn canopy doesn't always necessarily prevent all the sunlight from reaching the soil, and plenty of palmer surviving and going to seed that's not overtopping the corn as well. So considerations, if you see, chest-high plants of palmer and you're harvesting corn next year, or chest-high pigweed plants, might be palmer. Doesn't always have to be 13 feet tall and be quite as aggressive as we show with the shock and awe pictures. This particular cornfield also butted up against a soybean field. So similar to pictures from the south and southern areas of the country, can palmer grow taller than soybeans? Well, here's kind of the answer. Soybeans were planted all the way to that cornfield. You can see off to the right, the top right of the picture, where the soybeans are senescing is that brown color. You don't see that senescence in the foreground of the picture because the palmer is generally taller than the soybeans, so you just can't see the soybeans in this picture. So in this case, this was an area that cattle were fed on over winter, so a lot of the feed probably fell directly onto the soil. Also pretty high in nitrogen because of manure. So once we get into these more nutrient-rich soils, this is where palmer and other pigweeds like water hemp can really thrive and be as aggressive as we've seen from pictures from other states across the U.S. Again, I said we found this was related to cattle feed, this particular infestation. There's palmer on the left growing in a manure pile, so some mature plants on top of that pile. A little further down the pile, here's about a 3-inch tall palmer plant. This was the day after Labor Day where all these pictures I'm showing were taken. So just like water hemp, can we get season-long germination of palmer? Yes, we can get palmer germinating all throughout the year. As long as the seed is near the surface, it's not covered in shade, and we get a rainfall event, palmer, just like water hemp, can germinate and then go to seed within about a 3-week window. Now, I mentioned cattle feed a couple times. The instances that we found this year were related to sunflower screenings. Other sources of feed can also be introductions of palmer. I know in Wisconsin this year, they found cotton hulls, palmer coming in on cotton hulls. We've seen and showed pictures of contaminated hay bales in the past as well. This year, for us, it was sunflower screenings where we found palmer. And again, picture taken the day after Labor Day, there's palmer germinating right out of that pile of sunflower screenings on this operation. So how long into the year can palmer germinate in North Dakota? At least September 8th, or whatever that particular date was. So the next question we really need to ask is, well, a couple questions here. I was curious about the contamination level of these screenings we were dealing with. How much Palmer amaranth seed was coming in with this particular source of introduction? And then the other thing we need to ask is what type of herbicide resistance is coming in if it's coming in with some sunflower screenings. So to answer the first question, we went ahead and had some undergrads have a pretty good time of sifting through these sunflower screenings and counting out the individual pigweed seed. So how we went about this was we counted 10 different lots of 100 grams of the pure screenings, and 100 grams is roughly a quarter pound of screenings. The counts of pigweed seed, so again, this is just pigweed seed, just looking at the seed, we can't identify Palmer from red root or tumble pigweed. We found anywhere from about 940 to 1,700 pigweed seed per pound of screening. Average out over those 10 samples was about 1,300 pigweed seed per pound. Once we genetically tested and then also grew these out in the greenhouse, the vast majority, over 90% of those pigweed seeds were indeed Palmer amaranth. The rest were primarily red root pigweed, but we did find a couple tumble pigweed in here as well. So in the sample that we dealt with, a pretty high infestation level. That's not saying, you know, all the screenings shipped around the state that had infestation were that highly infested, but that's what we were able to deal with, and that's what we found in the screenings that we acquired. And here's just showing a picture of what else is coming in through those screenings. So here's planting a quarter pound of screenings, and most of what you see in that picture is Palmer amaranth. There's also some volunteer milo in there. There's a lamb's quarters if you look hard enough, and also some velvet leaf, which is not all that common across North Dakota, but the sites we were able to get out and walk, we also found velvet leaf in several of these field sites as well. So to put this in more of an economic perspective, the cost of the screenings, once you boil it down to how much does this flat of a quarter pound of these screenings cost, that's about what a half a penny could buy you. So when we talk about contaminated screenings, yes, they can be a good, cheap source, alternative source for feed. But if you're shipping a lot of Palmer amaranth, we kind of have to run some economics on controlling Palmer on the back end as well. A reason we're just kind of raising awareness this year. So the other thing I mentioned was screening this Palmer for herbicide resistance. What's coming in as far as resistance with Palmer in these sunflower screenings? So our first shot of this, we picked a couple of different modes of action, applied what we basically called a 1x and a 3x field rate on 3-inch tall Palmer in the greenhouse. So the herbicides in their treatment list is there on the screen. A quart of Roundup Power Max and 3 times that. We chose Raptor as our group 2 herbicide, Flexstar as our group 14, atrazine, and then we also looked at half a pound of 2,4-D and half a pound of dicamba. And I want to leave this slide up here for a minute or two because there's kind of a lot to digest. What I'm showing here on the screen is the percent of Palmer amaranth survivors 21 days after treatment. So I'm not showing percent control. What you're seeing is the percent of plants that survived that treatment. And this is 22 replications. So the bar on the far left is Roundup Power Max at a quart per acre. 55% of those plants survived that treatment. That 3x rate, 3 quarts per acre, we had 10% of Palmer plants survive that treatment. Moving over to Raptor, group 2 herbicide, whether it was the 1 or the 3x rate, about 80% of those plants survived. And the reason I'm showing survival is I'll tie this into resistance here in a little bit, but just showing that we have some variability across the different treatments. When it came to Flexstar in the middle part of the chart there, we had 1 survivor out of 44 reps across the 2 different rates. So kind of interesting coming from sunflowers. We would think some exposure, at least to Spartan, still very good control with Flexstar on these populations. Atrazine, about 50% of the plants survived or more. And then dicamba and 2,4-D were variable. But if we went to the higher rates, higher than we would use in a field, we did still have some survivors, but would they go on to produce seed? Most likely not in a field situation. So it's a couple pictures from this screening. Again, here's just Raptor untreated, 4 ounces of Raptor and then 12 ounces of Raptor. are we having some plants come in with a lot of resistance or high level resistance to group 2 herbicides and Raptor? The answer there is yes. But I said 80% survived. So we still had 20% of these plants that were killed off by either 4 or 12 ounces of Raptor. So something to consider. We talk about managing Palmer like you're always going to have group 2 and then glyphosate resistance. And population level, yes, but we're still going to control some of these plants. Typically, we'll go out and collect survivors in a situation like this, but kind of interesting and unique here that we were able to intercept plants on the front end of an introduction event rather than plants that went out into a field and survived a glyphosate or another herbicide application. Again, I mentioned most plants were kind of killed off by our half pound of dicamba or 2,4-D. The ones that did survive, most of them were curled down and probably would have died, but the occasional plant will look something like what I'm showing on the screen now. A little bit of injury, but overall survived quite well. That was dicamba. And here's the same thing with 2,4-D. So the other thing to consider is it's very difficult to do resistance testing on a population like this. And that's partly because we have a complete genetic crapshoot of what we're actually dealing with. When we're dealing with sunflower screenings, these are particulates that have been filtered out a couple of different times, and what's left over is just being sold for cattle feed. So there's no idea or no telling how many different fields or even how many different states from the country this population is coming in from. So we went ahead and did 22 reps, and we found a lot of variability with plants surviving or plants completely dying. So I've said we did 22 reps for glyphosate and group 2. Specifically, I wanted to expand that screen. And so we screened over 100 plants for glyphosate with the 1x rate. We also sent these off for genetic testing at the National Ag Genotyping Center. This testing will be available this upcoming summer. And basically what we can test for genetically if we have Palmer is the most common level of resistance or most common form of resistance, which is a higher copy number of EPSPS. So what we found with screening a larger number of plants is about 40% of these plants survived. Here's a picture of what this looks like. So again, do we have resistance at the population level? Yes. Is every plant resistant? No. Same thing with ALS herbicides. We screened a larger number. In this case, about 80% of those plants survived. And showing what that looks like. So more survival than glyphosate. Still have some plants dead in there, and then kind of a variability of what these survivors look like. Some highly resistant plants that were untouched, but also some that were pretty injured as well. So here's just kind of a quick text summary of this. I already have mentioned several of this, but based on our initial response and what we know about some of these herbicides, the glyphosate group 2 and group 5 resistance, I'm feeling pretty confident that we do have that coming in. The survivors there showed little symptomology. Dicamba and 2,4-D, they're concerning. That's a little bit more difficult to tease out, especially given a variable population. We'd have to go back and do some more resistance testing, comparing that to a sensitive. The good news is our group 14 herbicides still do seem to work on this population coming in. Going to finish up with 2 slides here, a selfless plug. I've gone off on a podcast adventure with my colleagues at K-State and University of Missouri. So if you want a podcast about everything weed science and things that take more time to talk about than what I can cover in a 20 or 30-minute talk like this, War Against Weeds podcast, episode 1 is posted. Episode 2 will be posted tomorrow, and we'll be dropping episodes every Wednesday throughout 2021. So episodes will be about a half hour long, just something fun to listen to, and pretty excited about that project. Otherwise, here's my contact information. Again, the QR code for the electronic version of the weed control guide. And I'm going to move here and put the QR code on the screen and then address these questions that have popped up in the chat. So before addressing the questions, for those who are done and want to scan the code and then move on with the rest of our day, really appreciate taking everyone's time this morning. Hopefully a lot of good information was obtained across all of our different talks. Please look for a survey we will be sending you tomorrow or the day after. We want to ask a couple follow-up questions about how you think the workshop went this year. One thing in particular is, next year, assuming we're allowed to meet in person again, would you like to see a hybrid component and be able to watch virtually again like you are now, or is everyone really geared up to come back in person? So we'll be sending a survey to your email address tomorrow or the day after. Please look for that, and please take a couple minutes to answer that as well. And then again, want to thank everyone for their attention. We will have these videos are recorded, and we will post them online, hopefully sometime early next week. If you want to go back and watch them again, any of the particular videos, if you logged in late and missed them, or just want to watch one again, they'll be available online. Again, should be starting next week. So thank you. And then I'll go ahead and answer these questions on the screen and make sure we have no lingering questions for any of the other presenters that might still be online. So let's see. Brad asked, is a hot, dry spring going to affect water hemp emergence? You know, Brad, with it being dry, most likely. With most water hemp emerging from the top half inch of the soil profile, if that is dry and not saturated, it's going to be difficult for water hemp to emerge. So if we get a shot of rain and saturate that top profile, good chance that we'll get emergence as soon as that happens. Philip asked, has it been considered to list water hemp as a noxious weed? Grant County has already done that. Several other counties in western North Dakota are evaluating the potential for that as well. In eastern North Dakota, where water hemp has been found, I don't think there's been much talk about making it noxious weed, simply because there's so much of it. It just wouldn't make sense to put that on the list at this point, as far as an enforceable weed for our weed control officers. And then David asked if Palmer is as salt tolerant as kochia. University of California has done this research, and if I remember the results from there, it is salt tolerant, but not as salt tolerant as kochia. So I don't think it's going to survive in the severe saline areas that we have, but it does have some salt tolerance compared to other pigweeds. So I think that's gotten through all the questions. Again, thank you, everyone. We'll cycle through these QR codes again, and hope everyone has the rest of their good day and a good week. So thank you again for attending.